All right, excellent. Thank you very much for having me. This is a great opportunity to show you what we're doing and just to regroup and talk about some new approaches to science. So I'm not going to talk for the whole time. I want to talk to the audience really and hear feedback and answer questions. So I'll probably talk for about half an hour, and then we have plenty of time to talk amongst ourselves. Okay, so there's a lot of history with all of this, and I've not been around obviously for all of it. So I'm not going to, I know the time zones are not perfect for everybody, so I'm not going to talk for half an hour about history. I'm going to get straight into what's new, because a lot of people, probably presumably most people that have logged in know something perhaps about spatiotemporal mapping. So basically, so we work on internal organs. We work on bladder and uterus and gut, some blood vessels, and there's a lot of techniques that are out there which I think can benefit from working together. And this is a good example I'm going to talk about in the next half an hour or so. So 20 odd years ago, 3 different groups more or less came out with a similar technique. So this is applied to the gut now. So Dr. Bachaucha's group in Paris published a paper about 21 years ago on showing spatiotemporal mapping in isolated segments of gut. Dr. Costa at Flinders University in Adelaide showed a very similar approach, and Premsor Bursek in Lucerne in Switzerland. So spatiotemporal mapping on its own is not new. What is new is what I'm going to talk about. So this is basically what it is. It's basically where you use some form of video recording. This is a picture taken from Michelle's paper, where you have a video recorder and a segment of gut. It doesn't have to be gut, it could be any moving tissue. And the changes in diameter of the gut are basically plotted as changes in grayscale. And it gives you a very nice high resolution vision of how events locally or propulsive are occurring over a segment of tissue. This is a figure from Marcello's paper with Grant about 22 years ago. Very similar thing, a video camera, computer or a VCR recorder in those days, and a segment of tissue. So for those that haven't perhaps done this sort of thing, when I started doing this and learnt about it from the experts, the problem with it was you make a recording and then you don't really have any idea how it went until you terminate the experiment and then play the experiment back and hope that you got some sort of artifact-free, clear recording. And often you didn't, because this is very much prone to light errors. So it became clear to us about 2-3 years ago that we needed some system where we could see the image plotted in real time during the experiment. That was the first thing. So it's a bit analogous to in a hospital, if someone needs an ECG recording, you don't want to make the recording from the heart and then play it back a few hours later and see what happened. You want dynamic output, so you want to know what's going on, and that's what we wanted. And it'll become clear in the next 10 minutes or so why we would want that. What's the point in having a real-time recording? So this is a segment of rabbit intestine, isolated from an animal, and you can see the movements are very complicated. There's lots of what we call phasic short bursts of activity here, and then underlying that there's another slower propulsive contraction. So this sort of tradition, what we would call peristalsis. So this is where spatiotemporal mapping really became very useful. So you could, as I said, plot changes in grayscale and reflect those as changes in diameter. So, so this is the same segment of gut now, just reorientated. If you look at the tissue, what we could do is see that the patterns of activity were not simple. And that was clear since Cannon 120 years ago. But what you could resolve was that there were distinct patterns. There were propulsive patterns, and then there were more non-propulsive phasic type activities. And as we became more and more curious about the mechanisms of the gut, we realized that it wasn't really good enough just to record motility. We knew we wanted to get a vision and a window into the circuits of the gut, and how were the patterns of activity in the GI tract actually responsible for these movements. So, you know, obviously the gut, the composition changes all the way through it. This is a segment of colon from a mouse. We've sped up the image considerably. And you can see now with solid, this has been known for about 45 years, that segments of content will be propelled in vitro. So what we've been very interested in is the mechanisms underlying this propulsion. And I can't emphasize enough, particularly to the early career scientists that are just embarking on a career, it's so important to understand mechanisms. Trying to cure disease before you understand the cause of the disease is perilous. And we do not know how the gut works. The heart, we have got a very good understanding. Drugs are very specific. Channels are very well understood in the heart. In the gut, we don't know that. And the gut's the largest internal organ, as you know, so we've got a lot to learn. So this is a still image of a spatiotemporal map, and all I just wanted you to know and see from this was those little fast contractions that you could see in the video before. That's these little white bands we call ripples that look like ripples on a beach. And then there was a slower pattern which you wouldn't be able to detect with the naked eye, these slow propulsive contractions which sweep down the gut. When we paralyze the nervous system with exomethonia, they terminate. So this is the sort of activity that's been around this is a paper we published about 10 years ago. You can record movements really from any organ, from any species. This is a fish. We took out some intestine a few years ago, and we could draw regions of interest and record spatiotemporal maps from fish intestine. So don't think the gut is the only organ that you can do this with. So this is some work we did about 10 years ago. John Arkwright's a physicist, and he made a fibre optic catheter, and we were able to insert it in the lumen, recording intraluminal pressure with spatiotemporal demapping with outflow along the gut wall. That was great, and we were very pleased with that. The problem was that you really need two recording devices. So we have a computer. Can you see my mouse there? Okay, so we have a computer for recording the demapping with the camera, and then we have the fibre optic recording completely independent. And that's suboptimal because it's very difficult to correlate perfectly in time the two recording systems. So we did use timing lights, which we've been using for a while. That's not particularly sophisticated. It was not the ideal system, although it worked. So what we've done, and this is really the essence of what I'm talking about now, is this a little video that Tim at Postdoc put together. This is our iMac. This is just what we would be recording in the lab. Over on the right-hand side here is our real-time software, which Luke has made for us. So here's the segment of gut. We have one computer now, one interface. We record extracellular recordings with 3 recording sites, which I'll talk about in a minute. We plug into an AC amplifier and a DC amplifier straight into the power lab. And so what we record in those red traces, this is 3 independent AC coupled recordings and the same channel in DC mode. So over here now is the spatiotemporal map playing, and we have global intraluminal pressure down here with the Miller sensors, intraluminal pressure local at 2 sites within the gut. Now, that'll all become clear why you would want to do that in a minute, but the most important thing for us is correlating in time the 2 events and seeing what's going on. It's impossible. Electrophysiology is difficult on its own with one recording site. With multiple sites and then trying to correlate that with movement is not advisable unless you can see the propulsion or motor activity taking place. And that's what Lauren and Marcello have been doing. So it's a much simpler system. It's not simpler in concept. The experiments are not necessarily simple. So we have a segment of tissue, whether it's uterus, bladder, heart, or guts, in vitro. And we have our custom-made extracellular recording electrodes. Now, these are really important that they can move. They don't, the guts not move. I can talk about this for 2 days if you want, but I'm sure you don't want to. The important thing with extracellular recordings is the electrode is adhering onto the serosa and moves with the tissue. So these are adhered to the serosa. We have the 2 intraluminal catheters, and then of course the webcam, all recording into, from a power lab, into an iMac running lab chart video capture. Now this is the plug-in through lab chart, through 80 Instruments Power Lab. I should say that 80 Instruments have not paid me to give this talk. This is equipment that we've tested. We've worked with 80 Instruments for about 10 years, testing a lot of devices, and we're endorsing it. We use it because it's good, but you know, feel free to ask us about that. We use iMacs for a number of reasons. They're preferable. You might be thinking, can you do all this on a PC? I'll get to that in a minute. The operating system, Yosemite, the newer versions are currently not compatible with that video capture, and we can talk about that in a minute. You can use PC, but it lacks the capabilities of a Mac, we've found. You need LabChart 7 Pro with the video capture extension. You can get that from the web. LabChart 8, and this is something we've been working with 80 Instruments on. LabChart 8 and Lightning on Mac currently don't have the video capture capabilities. Okay, so we use this pressure transducer catheter from 80 Instruments, the Miller. We found them very competent and very good for luminal recordings. Really any laboratory animal you could use these for. They're too big for blood vessels, but they're good for visceral organs. So this is the sort of things that we can do, which we never could do before, and it's given us a tremendous insight into mechanisms of the gut, which we could never do before. So what Lauren and Luke and Marcello have been doing is they combine the DMAP, so this is the spatiotemporal map, the grey scale. So the white in the background here is the contraction, and the dark is the dilatation. So this is the propulsive contraction running down the gut wall over here. Here's the cecum, proximal colon down into the distal gut. And what Lauren does is places the intraluminal Miller sensor within the gut and then locates the extracellular recording directly outside that same site. So now we get a reading of the intraluminal pressure at the same time as we've got the AC and DC coupled electrical recordings. And that doesn't prevent the propulsion of content down the gut. And this was really our dream, is to understand what's happening with the electrical activity during propulsion. So what are the benefits of live video recording? Couldn't you just do this with what was done before? The answer is no. And I'm not aware of anyone that's done this before. So with multi-site electrophysiology and multi-site pressure recordings, it gives us a tremendous opportunity to see what's really going on. 19 years ago, can't believe it's that long, I was lucky enough to be in North America, and we did this study where we took out a segment of gut with my former colleague and good friend, and we stretched the tissue and induced this ongoing pattern of electrical activity in the smooth muscle. We knew that because it was so robust, it must be important in GI physiology, but we had absolutely no idea really in the body what it was doing. And that was tremendously frustrating. So 19 years later, thanks to the approach I've just described, we've got a much better window into what's going on. So what Lauren does, here's this came out a couple of years ago. This dark traces the propulsion of the artificial fecal pellet, which can slide over the top of the Miller catheter. Okay, so we're not preventing expulsion of the artificial content. We record intraluminal pressure with the Miller sensor, and then we have our 2 extracellular recording electrodes here in red and blue, with the DMAP on top of that. And that gave us really a whole new vision. And what we found, to our excitement up here, just to cut a long story short, was the same pattern that we recorded in Reno 19 years earlier. We've never seen this before with any of the other approaches. So what that meant is that we could correlate what was this event actually doing, synchronized EJPs and IJPs. And when we knew there were literally tens of thousands of motor neurons firing simultaneously along the gut, it was particularly exciting to us to understand how this translated into movement. So here's the gut, as I was showing. Here's our 3 custom-made extracellular recordings, and here's the colon from these 3 sites here. I can talk more about this later on if you like. So this just shows a very similar thing with the different frequencies of activity along the proximal, mid and distal regions of gut. And we found probably the biggest misconception is that there's a distinct unified pattern of motility in the gut, even within the same species. Patterns of activity vary enormously, even when within the same region of the same animal, depending on the stimulus applied to it. So I'm not going to get into all that right now, but needless to say, with this approach, we've noticed a lot of activities with different frequencies in different regions, which we have uncovered and didn't realize were as complex as they are. So the pattern I just showed before, this can also be uncovered. We call these TNEs, transient neural events. And every time we found the electrical discharge in the red trace and the AC coupled electrophysiology from these 2 extracellular recordings, we found in green was the luminal pressure change. So this is from the luminal catheter with the pellet that was designed to propagate over the catheter. So we were not impeding or locking content in the gut wall. So this is now with a freer to move pellet, and we were able to correlate the different patterns which we were never able to do successfully in the paper in 2002, now with actually movement of content. And you could apply this to the heart or the bladder or the uterus. We've used the uterus, it works beautifully. So in the last sort of 20 minutes, what I'm going to talk about is just how useful the approach has been for us in the last 2 or 3 years. We did some imaging of the gut to cut a 18-year story into 2 sentences, and we found that there was a distinct population of activities in the GI tract. During propulsion, in other words, when there was a large contraction sweeping down the gut wall, I'll use my mouse over here, sorry. There were 2 real phases. There was a phase where the enteric nervous system was relatively desynchronized and the neurons were firing out of temporal synchronicity, and then they fired in large collective bursts around 2 hertz. But interestingly, the excitatory and inhibitory neurons were firing off at the same rate, about 2 hertz. So what does all that mean in the body? So if you look at a segment of gut, if you just watch this video, we infuse some fluid into the GI tract, it distends content, and at a certain threshold on the left-hand side, you'll see the gut starts to contract right there, and then the contraction sweeps down the bowel. Now, what we wanted to know was what was going on inside the muscle as this wave was sweeping down the GI tract. Was the velocity of propulsion determined by the velocity of the neural wave in the enteric nervous system, or was it a completely different phenomenon altogether? So what we started off doing, and this is using the approach I'm showing you, here's our custom-made extracellular recording electrodes. This is unpublished work, it's provisionally accepted, so no one would have seen this. These are 2 extracellular recording electrodes that are adhering to the serosa. And what we do is we fill the gut up slowly with fluid, and you can see when the electrodes are spaced very similar distance, the excitatory potentials are temporally locked. So the wave is not swept fully past the electrodes yet, nor got to the distal end, yet the 2 recordings are synchronized in time. You might be thinking, well, that's entirely logical. If the electrodes are so close together, you wouldn't really expect much difference. what about if you place one electrode up in the oral region of gut and the same electrode now down into the distal colon? That's separated by about 35,000 myenteric neurons. That's about 40 millimeters of colon in a 1-year-old mouse. So we're talking about tens of thousands of neurons here. Would this pattern still discharge at a similar rate, and would they be synchronized? So now we move to the same tissue. Here's the electrode now in the oral region and down in the distal region. And if you look at what happens, the two electrodes start off rather asynchronous. The wave hasn't started yet. There's no contraction at this point, so there's no propulsion. Now, if you look at the left-hand side, the gut's starting to contract right there. Yet look what's happening to the electrical activity. The EJPs are synchronized over 35,000 old myenteric neurons. So the contraction waves nowhere near got to the distal region, but the excitatory potentials are temporally correlated down here with those in the proximal region. So I'm not aware, so they become in phase for a few seconds, and then they become about 50 milliseconds slightly out of phase. Never before have I seen any smooth muscles that have showed a sub-action potential threshold voltage change temporally synchronized over such a large region of smooth muscle. So that told us immediately that the nervous system was synchronizing in time, even though the contraction wave hadn't got down there. And I can answer questions if you want to. Why is this discharging? Why is this not reaching threshold? You might want to ask that in a question, and I can answer it in a minute. Okay, so you can combine the video image with the electrophysiology and demapping concurrently. Luke, who is here with me, he wrote all the software, so you can correlate in time exactly where the contraction is happening. So the green line up the top shows the contraction wave is starting now. Here's the DMAP here, yet the electrical activities are synchronized completely over that spatial field. There's no way we would have been able to understand what's going on in this organ without doing this approach. So I'm worried about going too long. This is what the circuit we've now uncovered. Just briefly, so ascending excitation has been known for about 150 years, and descending inhibition. But what we've discovered is that actually at the same time the excitatory motor neurons are activated in the oral region of gut, they're actually activating inhibitory motor neurons at the same time, both oral and anal to the contraction wave. That might open up for a nice question at the end of it. How would that evolve like that? So why is it useful to have multi-site electrophysiology and pressure recordings during video imaging? We could talk for hours about this, but I want to just hit it on the head with the most important things. There's major regional differences along the gut. Okay, even over short distances, we've found different patterns of activity. As I said, depending on the conditions with which we stimulate and the luminal contents and whatever stimuli are applied. So it gives us an opportunity to correlate whether the similar or different patterns of electrical activity occur during the same sequential propulsive wave. So during one event, is it the same electrical activity? And how does it change from the activity that I just described to you? What happens when you put liquid, semi-solid or solid material? Do those patterns of synchronization still take place? And thanks to Lauren and Marcella with this approach, they've been able to uncover that this is a segment of guinea pig colon, I believe. And this is the natural expulsion of pellets showing on the spatiotemporal map. So as again, this is time on the x-axis, in proximal region up here, and distal column down the bottom. And the transient neural events that I've showed that we discovered in 2001, we found actually are very potently induced when the content is actually expelled from the gut wall. So there's multiple patterns of activity which occur over multiple different regions, which can be uncovered when we use this simultaneous approach of pressure electrophysiology and demapping. So there's no real limit to the length of intestine with which you can study. And there's no real limit technically to how many electrodes you could place along the gut. Commonly, Lauren uses 3 recordings, which are these 3 sites here. Again, red and blue is AC and DC coupling. But this is a segment of small intestine, and I just wanted to show you that it's very exciting to us to understand how the gut behaves from start to finish, and we can study the whole intestine in vitro. So these are the ripple patterns which have been described by many authors for years in the small intestine, which is a myogenic phenomenon, occurs at about 30 to 40 cycles per minute. And then once you get into the colon, in the proximal region, there's different frequencies of activity. And it's been really interesting to us to understand how the same, as I said, propulsive events recruit different frequencies of activity over regional differences. So this is just another example to show you of what we can do. So here's the green is the Miller sensor, the intraluminal catheter. And here's showing a larger burst of activity in the proximal region up near the cecum that's associated with the same propulsive event leading down into the distal colon. So the nice thing is that we can infuse content through the gut wall, which will propagate over this Miller sensor and not interfere with our DMAP, and yet still make the electrical recordings from the serosa. Okay, so how are we going for time? 2:30. I'll finish in a few minutes because I want to answer questions and talk to the people. So there's a great diversity of firing frequencies, smooth muscle action potentials, and there's a whole window. This is like being given vision to us. And people have recorded electrophysiology for centuries in AC and DC mode, but coupling it with the motility for us has been a major step forward. So I don't want to open up a whole new pattern of activity, but this is just a common sort of event that we could get from the small intestine and the colon. There's the different frequencies and different durations. This particular event here's the luminal pressure, which nicely matches with the bursts of DC and AC coupled electrical activity. And this is the underlying propulsive contraction underlying those events. Here's another event that's occurred locally, just to show you that not all events correlate. And this is picked up by the Miller sensor down in the distal region, which also obviously correlates with the patterns of activity, electroactivity. One of the interesting things that we've been able to pick up is the difference between propulsive and non-propulsive activities. There's a unique pattern in the colon. We have absolutely no idea what the clock is, but there's a fascinating pattern which is able to govern a propulsive and then non-propulsive or weakly propulsive event. And it'll sit in this oscillatory cyclic pattern for hours. So if you're interested in intraluminal pressure, the 2 Miller sensors works really well, coupling with the electrical and demapping, and they don't interfere with the propulsion of content. So here's the underlying DMAP showing propulsion. The other thing you might be interested in, not everybody studies the nervous system. There are many organs without. In fact, the GI tract's the only internal organ with its own nervous system. All the other organs haven't evolved with an intrinsic nervous system. And you can record myogenic activity. So the Miller sensors we've found to be quite productive. This is the AC coupled recording. This is the Miller sensor, and this is the DC electrical activity. So you can see that the flow of activity in these tubular preparations correlates nicely with the local, not global, changes in luminal pressure. Okay, I think that's the recording I just showed you. Okay, and that's another pattern of activity that we've often recorded, where we can record rhythmic bursts of This is a neurogenic phenomenon, and this has been of particular interest to us for beautiful patterns of activity to record in this era of functional genomics. We're really living in a renaissance era now, and applying the functional genomics with these approaches is gold. So you don't have to use electrophysiology or pressure. You can use, and people thought this was not possible, you can use simple mechanical recordings. This is a segment of mouse colon with a video camera. And what I was just showing you, you can use the video capture interface through 80 instruments, and you can record mechanical activity, which is in the white. So this is for recording, mechanical recordings from the mouse colon with the superimposed DMAP underneath. And this is actually the pattern I was just talking about. So here's a propulsive contraction that turned into non-propulsive or half the gut, and then generated this alternating pattern. So there are many, many derivatives to recording with this. You don't have to subscribe to what I'm telling you. You can make it fit to purpose for your organ and the aim of your research. We're interested in optogenetics and chemogenetics, and my good friends in St. Louis, Hong Chen, Jing, and Zilli are working on some great work at the moment. This is a study we did a few years ago. Very simple study with a laser, a genetically modified mouse, where we knock channel rhodopsin into the calretinal neurons. And this is the DMAP with motility. So the blue and red and green is mechanical activity. So this is a spontaneous contraction going down the small intestine of a mouse. And at this point here, that black, this black trace here is the artifact caused by the illumination of the light that induced the contraction, which actually propagated retrograde. So when orally and distally along the mouse small bowel. Okay, so 36 minutes. I think we've done pretty good. I know everybody's getting late in some countries. So what's the advantages and what's really new? I can tell you, after studying the gut for 23 years, we're really in an extremely exciting era, and we've uncovered so many things I never thought we would do. The advantages of live spatiotemporal imaging. If you've done spatiotemporal mapping, and then you realize the frustration of doing an experiment and then having to go home and find out that there was too much lighting in the room one day, which is very common, and the maps are all blurry, it's priceless to see a live feed out. So the patterns of activity, electrical activity in the smooth muscle of internal organs are complex. There's major advantages of live visualization of maps, spatiotemporal maps, and it's absolutely critical to have it live if you plan to do electrophysiology from even one side. I highly recommend you try multiple sites and with multiple pressure recordings. It's been extraordinarily insightful to us. And the direct temporal correlation of luminal pressure with electrophysiology during spatiotemporal map really allows us to understand how these corresponding propulsive movements link in time and what's the underlying phenomena that's responsible. So it also gives us a chance, as I said, to identify the regional differences in the patterns of activity along the smooth muscle, particularly during propulsion of liquid or solid content. So as I mentioned before, I think I talked about this already. This is really important. I've got Luke with me here and Lauren, if you want to ask questions about how you do this and why are these versions appropriate. But I can tell you they've spent a lot of time doing the work. I don't do the work, they do all the hard work and experiments. Definitely an IMAX preferable, as I said, with Yosemite. The newer versions are currently not compatible, okay? I would discourage a PC. I'm not sold on apples for everything by any means, but for this approach, I definitely think the apples are a better approach. LabChart 7 Pro with the video capture extension. And as I said, LabChart 8 and Lightning on Mac currently don't have the video capture capabilities. Okay, so the most important slide is Luke, who is our computer software genius. Not necessarily the most, but equal most important is Luke. Marcello has been the godfather. He's been at Flinders for nearly as long as I'm alive, which is nearly 5 decades. Lauren, who's done most of the electrophysiology and recordings you've just seen. Lee, who's done the imaging, and Tim, who's done many of these experiments, and he builds the custom-made extracellular recording electrodes, which are very, very useful. And David, of course, and Mel, and Kelsey. And the great people we work with overseas, Hong Chen-Hu, Jing Feng, Zili Ji, Rob Giroux, Malin Lagerstrom, Yves de Chronique, Marie-Yves Piquet, Brad Barth, Shilling Shen, Warren Grill, Yiwan Yang, Changsheng Wu, John Rogers, Zachary Knight, and Brooke Jarvie. So I think I'll stop there. Thank you very much to the funders that make our science possible. And I'm going to leave you with an important quote. I think the most important thing for a junior scientist is to ask the right questions. And John Tukey is the American mathematician who invented fast Fourier transform. He said an approximate answer to the right question is worth a great deal more than a precise answer to the wrong question. So I am going to stop there, and 2:40. We have time for a lot of questions, I hope. And I have Luke here that wrote the software, and Lauren that did a lot of the experiments, and we'd be thrilled to talk with you and learn from your experiences as well in science.
Thank you very much, Nick. Just be waiting on questions in the Q&A or the chat.
Helena, that's good. Nice to see you here. Helena has raised her hand. How do we hear Helena's question?
She can just type it in the chat, or she can just type something in the Q&A. Okay, here we go. We've got something from Chelsea Lee. Incredible work. Would attaching the electrodes / sensors on the PrEP affect motility / electrical activity? Example, triggered contractions due to the mechanical receptors in the gut.
Good question. I can't see that question. Did you just read that out?
Yeah, I did. Under the question.
I think I got the gist of it. It's a very good question. And yes, recordings can stimulate the gut. It's an unavoidable consequence of the recording. The gut is extremely sensitive. It's a good question. And the answer is yes. Recordings do stimulate the gut. So we do minimize and pretty much eliminate, I would go almost as far as to say, the potential for that by having prolonged recordings with gentle suction applied to the serosa. So it's very, very clear if you stretch the gut's very, to me, it's very much like the heart, a bit like Frank Starling's law of the heart. The more you stretch it, the more forceful it contracts back. So, I mean, obviously we're trying to understand the mechanical activation of it. So when we're inducing distension, we actually, of course, we want that pattern to be induced. I think your good question is, does the recording itself modify or induce patterns that might lead to spurious results? And acutely, yes. So if you were to lower your electrodes immediately onto the gut or rapidly change resting tension, it definitely will induce patterns of activity. We don't see, over prolonged periods of time, we don't see any evidence of that. It's a little bit like, if you imagine the knee-jerk reflex in human, if you apply a rapid force to your patellar tendon, your knee will fly up in the air. If you apply the same force over a prolonged period of time, your knee won't respond. So it's a matter of minimizing the stimulus, and you can minimize the artifact.
Thank you, Nick. Helena Parkinson has a question. Brilliant. are those small electrical oscillations associated with small oscillations in tension, just local, not involving the entire circumference of the tube segment?
Good question. I think I know the image that you're talking about. I don't know if that particular, normally if there's a neural phenomenon, the answer is yes. The voltage change is synchronized by transmitter release from the motor neurons right around the gut wall. That particular event, I think the one you're talking about was myogenic, and I'm a bit hesitant to say with confidence whether they're perfectly synchronized right around the circumference or not. We would need to do that in the sheet preparation and record from each circumferential edge, which I don't think we've done. So I'm sort of guessing with that. I would take a punt. As you know, also a lot about slow wave Solana and recording from smooth muscle for many years. They do discharge those events in the gut, at least in both muscle layers. So I need to be sort of guarded whether or not it's just a circular phenomenon or it's got some mechanical interference from longitudinal muscle as well.
Thank you, Nick. We have another question in the Q&A by Weifeng Rong. Thank you for the very informative talk, Nick. What's the tip size of electrodes for electrical recording? Do you use thick-walled or thin-walled glass pipettes?
Good question. That's right into specifics, Lauren. So Lauren is here. She's telling me thin-walled glass. Yeah, I think 1 mil diameter. 1 mil diameter.
0.75 internal, I think.
The current feeling is 0.75 mil internal. So we can confirm that back with you, and maybe we could take a photo and send you the dimensions. I mean, they've been specially crafted for small laboratory animals, but you know, which I guess would be suitable for your research too, knowing the tissues you work on. But they can be modified, of course. But I'll get a photo and we'll take it and send it to you.
Thank you, Nick. Okay, we have another question in the Q&A by Sasharia Karabu. I apologize if I pronounced that wrong. Nice talk, Nick. I was wondering whether the burst type activity in the electrical recordings are spike bursts thought to be calcium currents in the smooth muscle, or is it a separate activity occurring in the neurons?
Okay, so well, actually I can go back to the slides. So this particular pattern, so in smooth muscle, in fact, in almost all smooth muscle that I'm aware of, the primary inward currents due to calcium, there are definitely some smooth muscles with a TTX sensitive sodium current, and Helena, who asked one of the questions, has published that. But nifedipine or any of the calcium channel blockers usually knocks out the inward calcium current in the muscle. In the nervous system, we have very little. I'm trying to find the image that you might be thinking about. In the nervous system here, this is actually subthreshold. So these are calcium transients, if I'm answering the right question. This is in nifedipine, where we've paralyzed the muscle so we can immobilize it. There's not a lot, there is some evidence of a nifedipine sensitive calcium current in enteric neurons, but it's weak. Most of the action potential in nerves, in vertebrates, is inward sodium. But there is definitely a component of calcium. So these are actually, this is actually a little bit more complicated. So we've paralyzed the muscle with nifedipine, but we haven't paralyzed the nervous system. And using flow, the kinetics of flow is slow enough. I'm not sure if that's the right terminology to use, but it's too slow to capture an inward current, any current. So the activation kinetics of this calcium indicator is such that every time you get if you were to record from these neurons with electrophysiology, the spike would be occurring probably about 70 milliseconds before these calcium transients. So we're talking about hundreds of transients across, literally thousands of neurons. So this is second messenger. I'm not sure we want to get into all this, but this is second messenger driven calcium in the cell bodies by the inward sodium and or calcium current. So the calcium indicator that we use doesn't have the kinetics to capture the spike. Some people have done that, but spatial field is down to about one cell body because you need about 2 kilohertz acquisition rate. And we're not interested in capturing that rate. We were interested in large spatial fields and how network behaviors were activated with tens of thousands of neurons. So I'm not sure if I hope I've answered the question properly there.
We have a question from Almar Al-Abed. Thanks for the great presentation, Nick. In terms of synchronizing the electrophys and video capture, how much of what was in-house programming versus AD Instruments video capture plugin?
Good question. Luke is my computer whiz. Did you read that, Luke? In terms of synchronizing the electrophys and video capture, how much of that was in-house programming versus AD Instruments video capture plugins? So,
in terms of synchronization, all of it was in-house programming. In terms of actual lights, the lights from the pressure catheter, the multi-sensor pressure catheter, was based on their own hardware. But in terms of synchronization, once we've videoed the gut, we could identify manually regions where the light shows up on the video, and that the time when a synchronization pulse is sent and recorded on the lab chart, we know when that occurs, and we can see that light flash in the video, and so we can correlate those two pulses.
Okay, thank you very much for that answer. We have another question here by an anonymous attendee. They ask, "Hi Nick, really cool work. This is a little off topic, but we do a lot of ischemia reperfusion injury in the gut and frequently see the most injured areas of the gut are where content is present. Do you think this could be caused by increased activity in these regions?"
By the gut being in those regions? I mean, the content being in those regions, is that the question?
I believe it is.
If you could just please read that last sentence. I think I got what the question was.
Sure. We do a lot of ischemia reperfusion injury in the gut and frequently see the most injured areas of gut are where content is present. Do you think this could be caused by increased activity in these regions?
When you mean increased activity, you mean increased activity in what? Increased in the muscle, mobility? I'm not quite sure. If that's the question, my simple answer is that I don't know. I don't know a lot about ischemia. I mean, I'm not sure how that induction is taking place. Often, I mean, what I'm thinking of is ileus, very commonly in surgery in human patients. A large proportion after the bowel's been handled, they develop inflammation and the gut becomes completely inactive. And that's a mystery to us, whether it's inflammatory cytokines and so forth. So my answer is I don't know. It's a good question and I'm really just guessing. But my suspicion is it might have something to do with the release of cytokines and inflammatory mediators at that site. I don't know what what the content at that site would be contributing to that phenotype. It's a good question, though.
Thank you very much, Nick. If you don't mind, if I could just present the last slide with some contact details.
Yeah, sure. Yeah.
We'll have one more question after this.
Sharing.
Okay, we do have one more question here by Benjamin Hoffman. Sorry. Nice presentation, Nick. Are you able to do any of this work in vivo versus isolated gut? Have you also looked at how a healthy gut compares to a diseased gut?
Good question. So the fiber optic catheter, high resolution, that was made by John Arkwright was tested. So we did a lot of the testing here over the last decade in animal models, but it's gone, that catheter has been used in human. And we have done a study recording in high resolution from human conscious in vivo patients and then removing that disease segment of same colon and recording from it with these approaches in vitro. It's not that easy to do demapping or videoing extraluminal in vivo. So you can place a camera, obviously a colonoscope, within the gut wall, like as in a colonoscopy. So we can get a movement inside the lumen, and that's actually something that we've been trying to do. Marcello's been working on recording motility via the mucosa, and that's been very interesting. So we can do that conceptually. The electrophysiology would be tough. We haven't done it. I don't know if we'd get ethics for that. Our good friends in New Zealand are doing this in human stomach. So the patients anaesthetised and they record extracellular across the gastric wall, and I believe recording motility as well. So it can be done, but it would be a toned down derivative of it. And the last part of the question about was disease gut, I think.
Yeah, that's right.
We have a paper which I can send you where we recorded from 4 patients in vivo that had intractable slow transit constipation, and the gut transit had been basically there was no transit. So these patients were last resort. So we got an opportunity to record from them in vivo, as I mentioned before, and then take that exact same whole colon out and record from that tissue out of the body in an organ bath. So now it's away from any extrinsic nerves or hormones. And the patterns of activity that were in vivo to in vitro, it's a tremendously interesting field of science. It's unfortunately very limited in that, of course, we never really get healthy human gut to study. So we always have that disease state. But our quasi-healthy state is the tissue that's taken the margin away from the tumour or whatever the lesion is. So there's a few caveats with human work. We have done some with impaired transit, but it's perilous. And the last year with COVID has almost completely terminated human studies. So we're hoping to get back into that.
Thank you very much, Nick. We'll get a few 30 more seconds to see if anyone else has any questions, but this is obviously being recorded, and we will send this to all the attendees today with also just some key contacts if you want to contact Nick Spencer or anyone from the ADI team. Yeah, it's pretty much done. I think that's it, Nick. So
no worries.
Thank you so much for your presentation today. It's very informative.
Oh,
and
great.
It's very pleasing to see that it has a lot of interaction there, a lot of people asking plenty of questions. So thank you so much.
Definitely. Thank you for having me. It's great and good questions.
Wonderful. Okay, we'll end it off today. So Thank you very much. Okay, bye-bye.
Take care.